Hello everyone, it's Dr. Wolfe here, and in this video I'm going to be going over how we could apply basic mathematical analysis to the data that we've collected, specifically for our bar graph. If we're trying to see the difference between two groups or to know if there's an actual difference or not, we can perform something called a t-test. So that's what this is going to be on. So let's say there's an experiment, and we've run this experiment, and it's the effect of light color on tomato plant growth. On the y-axis is the height of the plant, and then here we have sunlight versus red light. And it's clear that the bar for red light is bigger, right? 6 centimeters compared to 5. So the question is, did red light have an effect on plant growth, right? It's bigger, but is that much of a difference, right? I bet if we were to poll the class, half of the students would say, no, that's really not a difference, 1 centimeter. And others would say, no, that looks like a difference. So who's correct? And we can't just leave it up to it seems like it's different. We could actually run a test and be more confident that there's either a difference or there's no difference. All right, so that's the purpose of running a t-test. So in science, we want to be at least 95% certain, all right, that the averages of a control group and experimental groups are different. All right, so what we're going to do is from a t-test, it's weird, from a t-test we calculate a p-value, and we want that p-value to be less than 0.05. Now that 0.05 is actually a percentage, right? So if you move the decimal place, 1, 2, that's 5%. All right, so we want to be 95% certain, so that's our percent certainty plus this 0.05, that equals 100%. So we want our number to be less than 0.05. All right, again, the t-test is going to analyze if there's a difference between the control group and the experimental group to see if they're actually statistically different or not. Okay, so let's actually look at an example. So here we have an experiment. It's the same one we looked at the graph before, effect of light color on height of plant. And here we have one experiment by Don and Dave and the exact same experiment by Abby and Nikki. And notice that they got the exact same results that we had in our graph, averages of 5 and 6 in terms of the centimeters that the plants grew in both groups. All right. However, if we look at the data, the 10 different trials they used to get there, it's different, right? Here they're kind of close together to that 5, 4, 5, 6, and then to the 6, 5, 6, 7, really close to that average. And here it's all over the place, right? 1, 2, 10, 8, 4, 3, 7. Red light, 6, 12, 3, 3, 10. It's all over the place to get the averages of 5 and 6. So same exact averages here, but how we got them is different, all right? So we're wondering, we're still trying to wonder, are these 5 and 6, is that actually, is there an actual difference there, or is that due to chance? Maybe this kind of looks like it might be due to chance, or the numbers are over the place. But again, we can't just be like, oh, it seems like it, right? We have to run a test. So we're going to click on this link right here, the t-test calculator. It's going to bring you to this website. By the way, if we were to continue to go through these slides here, it would show you how to do it, but you could always watch this video for that too, right? So when we get here, a lot of the stuff is automatically going to be selected for you. Enter up to 50 rows should already be selected. We're going to click on an unpaired t-test if it's not already there. All right, you do not need to know the difference between these other things at all. All right, and then we're going to enter in our data. So I'm entering the data in for Don and Dave right here. All right, and just for the sake of time, I've already entered it in. And after that, I just click calculate. All right, now by the way, sunlight, right, in this group right here, group 2, red light only, data right here. Okay, and notice I did not put in the averages. So now I put in just the actual trials. I'm going to calculate, and there's a bunch of stuff that's going to come up. All you need to worry about is this number right here. The p-value equals 0.035. And remember that p-value is a percentage. And so if we move the decimal place to the right, 1, 2, that means that that's a 1.35%. So what does that mean? Let's dig into that. All right. Now, by the way, for the sake of time, I am not going to enter in Abby and Nick's data, but I would do the exact same thing. I would go back here. I would enter in those data, just the trials, not the averages. All right, click calculate now. All right, and by the way, we would get the p-value. We just calculated this, right, 0.0135 for Don and Dave. And if we were to do Abby and Nikki, we would get this 0.5045, right? Remember, this is a decimal, so move it 2 places to the right, and that's 50.45% compared to that 1.35%. Again, what does that mean? So that's what we're going to dig into now. All right. So how would we reach a conclusion based on these p-values? Remember, in science, we want to be 95% confident our results are not due to chance. All right. Here's an example of chance. Maybe I flip a coin 10 times. and maybe 5 times I hit heads. Maybe I do it another 10 times, and 2 times I actually hit heads, right? 8 times I hit tails, all right? That's very possible, right? Even though we know it's a 50-50 chance, sometimes if you only have so many trials, right, you could actually get different results. So that difference between the 5 in one group and the 2 in the other group, we know that that's just due to chance. There's a difference there, totally due to chance. We want to make sure our data is not due to chance. So Don and Dave, their p-value equals 0.0135. Remember, that's a decimal, so 1.35%. What that means is that there is a 1.35% chance, all right, that the difference that we see between that 5 and 6 is actually due to chance. So that's pretty good, right? We could almost flip that around, and we could say, okay, here, let's say 100% minus that 1.35%, that equals 98.65%. That actually gives us our confidence, all right? So this bottom part right here, the experimenters are 98.65% confident that the plants grow taller in red light compared to sunlight, all right? So it's kind of just a different way to explain it. So if I were to write a conclusion on this, all right, I would say that the p-value equals, and then I would state my p-value, and if it's less than 0.05, right, less than that 5% right here, I would say that those results are significant, that the difference is significant. If it's greater than 0.05, if it is 0.055, all right, that's too much, all right? And we would say that the color of light does not significantly affect, right? In other words, there's a difference, but that difference really isn't significant, all right? So here I'm going to say that the color of light significantly altered the height of the plants. The experimenters are 98.65% confident that plants grow taller in red light compared to sunlight. All right. So now instead of just looking at my data and saying like, yep, it looks like there's a difference, I have a statistical way to be more confident in saying that there is or not. All right. So let's look at Abby and Nikki. Remember, we didn't actually calculate this, but if we were to, the p-value would be 0.5045. Remember, this is a decimal, so I'm going to move it 1, 2. And so that means that their results were 5 and 6, right? And so is there a difference there? Well, it's actually about a 50% chance that 5 and 6 is just due to chance. That's really not like good odds in terms of like, oh yeah, the red light definitely had an effect. All right. So if I were to write a conclusion, all right, because this is greater than 5%, this is greater than 0.05, I would say the color of light does not significantly alter the height of plants. There's a difference, but it's not a significant difference. All right. And I would say the experimenters are only 49.55% confident that plants grow taller in red light compared to sunlight. All right. So hopefully this video helps in terms of introducing p-values, why they're helpful, why they're important.